So this is unlisted. So if we get botted again, it's definitely somebody in our class doing it. Then we know for a fact, but it's not somebody who Googled it. All right, let me. And yeah, so yeah, so I'm old. We use YouTube. Oh, hello. Is that working? Oh, right. There was this. All right. Okay. I want to have that up. No, don't. Cripes. I hope nobody sees that. Because that'll give away the answer to one of the questions. All right, we're gonna start at 7:05 because never people never show up. But the other thing is, I would like to go to the bathroom before everything starts. So we have 38 questions. Most of them are 30 seconds each. One is 120 seconds. So it is a math problem. No, we shouldn't. We shouldn't math problem tomorrow. It is. It is written. Yeah. So the upperclassmen or repeat or whatever you want to call them, as I've gotten sources from both. And it says that is number 5 4, number 6. You do not get the which makes sense. And so that is number 5 and 6. No, it's 5 4 6. That's 5 4 6. Yes. It should be like glutamic acid. So yeah. So I did a 2-part question based off of number 1, and then I just did the one that we're having tonight. Is number 2. So you know how I'm doing it. Isn't it the one that they said on the no, because I didn't have enough time to put it in there. I was just wondering, big number 5 because I have not. I'll be very honest. I have not actually done number 5. I haven't done it either. I got up to number 3. So exactly how to do it in class, or is it a different one? Yeah. So she has. Yeah. So she has a video. She has like multiple videos, and she's walking through it. All of it. No, just number 5. Just number 5, and that's what people were telling me is that that's why number 5. Explain number 5 super in depth, so that you know how to do it. So like pretty much, you're an idiot. No, number 5. I mean, you didn't study, so okay, that's fine. I can do that. So yeah, if you can do number 5, you're fine. You're golden. Golden. Screw it. 10 points. Right. Yeah, right. Yeah. I thought when people were saying it was all or nothing, I can't. You did do all or nothing last year and the year before. So I heard last year she did partial. Last year she did partial. E-reads had to do. If you got one part wrong, you got it all wrong. But I guess probably got a lot of complaints about that. Yeah. You know why you get that all wrong? Because you killed the patient. You can't kill a patient. You kill the patient, you lose. Nobody ever is gonna have to do this unless you work. That's what I want to do. Oh, yes, go working on it. Well,
Alright,
I'm
then we'll
good
have to
though.
do what we have to do. Yeah.
Oh my god, I can't, I can't.
Yeah, same, same.
That makes it worse, yeah.
Yeah, it's like, oh no, don't. Man, hope you have a laxative.
Oh, the father. So. We don't. Got it. We got to do it. Let's have a review session. Oh, yeah. Yeah, put it tomorrow and we can, Thursday, Thursday and Friday as well. Yeah. What?
There's deoxy. What's the sorbitol mineral? What is it called? Oh, the alcohol sugars, deoxy sugars. You know, yeah. Yeah, that's possible.
That's for amino, deoxy, and alcohol.
And these are, that's gonna be like, which one is not? She has a, that's a, that's a.
Yeah. I know.
Rice and salad. Mix it all together. Makes it pretty good. I'm getting weird looks. What?
I didn't know you had rice in there, what?
Yeah, there's rice in the bottom. It's like sweet rice. Pretty good. With the rice.
Yeah, so you guys, I actually collected a microphone, so they might be able to actually hear us this time, so if we have to explain anything. The other thing is Casey said that there were mistakes in questions, but then Dr. Lee started denying them back about what
I don't know
questions.
mistakes, but she's like, yeah, I did correct it. That's cool.
Yeah, so,
She asked me if I was gonna yeah, she's like, they told me to look over the questions, and she's like, fix a few. I'm like, okay.
You should just email it back though, because then we're.
So we don't have the correct information.
So
I don't know if they were like wrong,
Like
like
wording.
wording,
So there's
like, yeah, wording, like maybe
Yeah.
it's too easy, too hard, like that's what I'm thinking.
Yeah, so there were a couple that like, the person who did polypeptides put in a lot of carbs. So, about half of them are like medium and then the other half are hard, and they did like 14. But I was fine with that because, A, that's our quiz tomorrow, so I'm perfectly fine with it. I did a couple of questions at the end that are more therapeutics questions, and I thought that like there's like one or two therapeutics questions, so you have to actually think and use like real world applications and stuff. Those are mine. But those are like the ones that I can see right off the bat that you guys are gonna. No, I didn't give her mine, but I know which ones she probably corrected. And then there were like spelling mistakes and stuff, so I fixed those. But we're gonna wait 5 minutes because 5 to 7 minutes because there's always you guys are never on time. You guys are never on time. You're close. You're close. But it's late. You guys have been here for 8 hours. I've been here forever because I live here now. The more I live at East Apartments, and I have a window that overlooks the parking lot, the entrance to the pharmacy school, and I see the pharmacy school sign every day when I wake up in the morning. And it's just like, God, I can't escape it. Like sitting on the weekend studying on my own, and I'm like, I just saw so and so walk in the building. Like, feel guilty. I haven't studied at all. Yeah, like I definitely could have done better this weekend, but I got hit by the apathy very hard. So there are questions in the lipids section, but I'll help you guys through them.
Exactly, that's what I'm saying.
Okay.
You have to deal with shit all the time.
No, that's not what I want.
That's scheduled. Get used to it at that time. Don't get used to it. I'm a grandma. I'm the old one. I deal with 17-year-olds.
Zero people who make your questions. Go blame them. No, no. I did a bunch of questions too, so I guess you could blame me. Takes a village to make us all pass school. I don't know if you guys, so we were talking earlier, I think Everett and I were, but, talked to somebody, the other thing that they had was that she also had to use how to do it. Explain this. I don't think a lot of people work out, but she doesn't just memorize. So this one has 7.2. Yeah, I think this one is 7.2. Right. But the other thing is like number 1 to Oh, yeah. So if I was a professor, I would like send half of those. Screw with you guys a little bit. Sure would be. Yeah. Well, because I want to see if you guys, if I was teaching this, if I'm putting myself in, if I was teaching, I would like to see that you guys do how to do this. So if I can like change a number and you guys still get it. Whereas if you don't change a number and do it straight off, then I can't get it.
The way you said that makes it sound like you're gonna get a chicken and then it. Now I'm gonna name my chicken the fish.
Damn it, still on my screen. No, what? Okay. You enjoy values in one is the two where like, oh no, did I? What? Your name? I was like, how do I have Oh, no. Okay. Billy Dilly. Oh my god. I say Everett's up there. That's your good friend. No, I have no idea what's going on.
So this is for the exam.
Yeah, so
Yes,
now
now.
we have a few big questions, and I made them super. The person who did it was a really good job, but because like I made it super easy, and if you guys start with us, I can like tell you guys the answer. Because I thought for some reason in my mind I thought that you guys had encountered maybe some of the stuff in like biology, maybe you'll know it. Yeah, like so. So maybe you'll know it, maybe you won't. But there's pictures.
I mean,
Got you on that.
We'll see.
how is it easy for them? I don't know.
I want to trash someone. I want to see exactly. I want to be super great at it and see how much of an actor I
All right. As much as I love you guys, your names are taking up half the screen.
That's why I'm going for you guys to know. So you realize how awful the person is.
Yeah. So somebody told me that they nominated me. And I had to like email Bureau, and I was like, no, no. I am an absolute tyrant, and if you want my ego to get bigger than this room, it's already quite large, and I can't get it through the door. So. All right, so 1, 2, 4, 5. So we have one person, maybe two people who are actually streaming. So that's nice. All right, so we are 5 minutes after 7, so we're gonna start. That is a quote from Salvador Dali. It says, "I don't do drugs, I am drugs." So, what is the primary protein structure? The Kahoot game pins at the bottom. So. Oh, ha. There. Uh-huh. There, now you can see it. Now it's at the bottom. All right. So, what are your part of your protein structure is your alpha helices? Secondary beta strands? Secondary. How about your amino acid sequence? Yep, that's your little ball model. And then you have a globular or a barrel. That is your third. So what if I, well, So, oh, well, okay, I spoiled this. So beta sheets are a blank structure. Are they a strong structure, a weak structure, a parallel structure, or are they not any of that? Okay, so beta sheets, because they have the hydrogen bonds, I believe. Yeah, so they have hydrogen bonds in them, and if you take like two pieces of glass and you put like water in between it, they stick together really, really well. That's kind of how hydrogen bonds are. And that's kind of how the beta sheets are. Now, beta sheets can be parallel or anti-parallel, so they can be both. But they are not considered a parallel structure. So this one was a little bit of a trick question. So I don't fault you, people who chose that or none of the above. The person who did the proteins did a lot of hard questions. So they did a lot of easy questions and medium questions, but then they did a lot of hard questions. So what is an example of a structural protein? So do you remember what some of these proteins are? Where do they function in the body? What do they do? All right, so collagen is a structural protein. So glutamine is just an amino acid, I believe, by itself. So it's not anything. It's not a structure. Collagen looks like a triple rope. And so collagen framework makes up your tissues, it makes up your liver, it makes up, so we've actually 3D printed a bladder and transplanted that into somebody, and they just 3D printed on collagen. So it's pretty cool. Albumin, where's albumin? You guys know? It's in your blood. So a lot of albumin, so that's where that is. Insulin, that is your pancreas. Yes, it is yes. So it's the stuff that your pancreas secretes. Matt, quit winning. No, you're doing work. Great. Beta sheets can be lipophilic, amphoteric, amphiphilic, or amphipathic. So what are the definitions of these? I have to double check this one because yeah, I think that this is polar and nonpolar. So oh.
That's what acidic and basic. I don't know the basic.
Yeah.
What I said.
Okay.
The one I said was amphiphilic. That's that one. The one that's like non-polar and hydrophilic. Take back what I said.
Empathic means that it is hydrophobic and
hydrophobic.
Yeah. So
water.
So the big thing that I can think of off the top of my head that has beta sheets that have this is that you have your sodium potassium pumps, and you have these in the heart. And so on the inside of the pump, it has these beta sheets, and then it has alpha helices on the outside. So the beta sheet needs to go through the lipid membrane. So it has to go through a hydrophilic, hydrophobic, hydrophilic area. And so the beta sheet can do that. So it's one of those interesting things. What is the most effective buffer for a solution that has a pKa of 6.3? So when you're looking for a buffer, what are you looking for in a buffer when you're looking for a buffer? Okay. So, Everett, since you went yes, you want to explain this one?
It's in within
No.
one.
Yeah, that is within one.
Yep.
Oh,
That's
shit.
the only one.
Oh, yeah, that's all. 5 is 1 point. Yeah. I didn't go with it.
Yep. So it's 1.05. The other one is, I believe, 1.3. So, and then the other one's way off, and then that leaves that last one.
It's good that no one put it on.
Yeah. Yeah.
I think everyone knew this.
Well, so 7.6, I think that there was a question in the slides, and the answer was 7.6. And so I put that down like out of instinct. And then I was like, later I went, wait a minute, no. You know? So, JW taking the lead. Matt. Oh, it's okay. I'll just pick on you. All right. If a functional group with a 7 pKa is put into a solution with a pH of 5, it is in equilibrium, protonated, deprotonated, or technical disequilibrium. So, all right. So what does in equilibrium mean? It means that the pH and the pKa are the same. Yep. So you have to think about this as an inverse, kind of a bit. This is how this helps me. So when the pH is low, that means that there's a lot of protons around. So if there's a ton of protons around when the pH is low, then that means that there's enough protons to protonate your chemical. And so if your pK is higher than that, then there's definitely enough protons sitting around. So, and this is one that I'm gonna beat into your guys' head a little bit because I think that there's like 4 questions like this. Hey, JW, Canada Dry is getting up there. Good job, Canada Dry. Which of the following is a proton acceptor? Acetic acid, sodium hydroxide, ammonium, or vinegar?
I was gonna say.
Yep, there are multiple right answers. So why is acetic acid and vinegar not on there? Because they're both acetic acid. So vinegar is 5% acetic acid. That's what it is. So, hey, you are on a streak. You are, in fact, on a streak. All right. So if a functional group is in equilibrium, that means there are some super simple questions. So just skip. You guys don't need to know. All right. Awesome. JW for the win. All right. What is an example of a structural protein? I thought I did. What the heck? All right. Why are we have? Well, there's different answers in the bottom. I have a feeling. Yeah. Okay. So collagen is again. Myosin is your muscles, and hemoglobin is a transport protein. So myosin is considered another I think it's considered another transport protein, or is it a moving protein? I can't remember what it's called, but it is in fact your muscles. What is the correct term for a protein made up of 2 subunits that are the same? So is it a monomer, dimer, heterodimer, or homodimer? I'm not sure if this one, yeah, this one had double answer. So what is a dimer? Dimer. Yeah, so di, two, hetero, two different, homo, same. Learn your Latin, people. We need to teach more Latin in school. No, no, we don't. It's boring. All right, so what is the function of a structural protein? So, you know this by now. Okay, well, so does it defend your cell?
I think I got it right.
Okay, does it help strengthen tissues? Does it store molecules? Or does it control other processes in the body? Okay, so structural protein is collagen kind of stuff. So, what is the function of a regulatory protein? So, what does that do? Does it catalyze your reactions? Does it control other processes in the body? Does it store other molecules or atoms for later use in the body? Does it defend against infectious agents? Got almost all the answers. And so, yeah. The regulatory protein regulates stuff that's going on in the body. So, enzymes catalyze stuff. Storage proteins, they're the ones that do. And then there's defensins. So, what is an example of a transport protein? Select all that apply. So this one is one that is multiple. So there are multiple answers to this one. So that's why I put that select all that apply, because I didn't want to like have people freaking out on me. Forgot to do that earlier. HDL lipoprotein. So it can be a transport protein, but not usually. And I think somebody pulled that from the slide wrong, but it is considered a transport on the slide.
Which one?
High density lipoprotein. So, okay. I wasn't sure about that one, but this person gave me last minute. I can't remember if they gave me last minute questions or if this was the person. What is an example of a protective protein? So, and this is one that also has multiple. So, fibrin is one that goes over and causes your scabs, helps you not bleed out, helps you protects you from more bacteria going in instead of just having an open wound. Immunoglobulin helps bind to like bacterium and stuff. And then your thrombin is also, I believe, a clotting factor. So that also helps protect from bacteria coming in and stuff. Bond responsible for connecting subunits in the quaternary structure. So what is that bond that is this one? If I remember correctly, this one's a little bit harder because this is one that is not between amino acids, if I remember correctly. But I may be wrong. Yeah. So in insulin, you'll have a disulfide bond, and in a lot of quaternary structures, you'll have covalent bonds. So, all right, true or false? Alpha helices and beta sheets can be polar or nonpolar, but not both. Yeah, 50-50 shot on this one. Okay, so we talked about this earlier that I believe a beta sheet can be hydrophilic and hydrophobic, and so you can also have polar and nonpolar at the same time because you have that hydrophilic, hydrophobic areas, and those are usually correlated with what's polar and what's not polar. All right, alpha helices can be aliphatic, but beta sheets cannot. And if I mispronounce that, I'm sorry. I'm an uncultured fool. So what is aliphatic mean? Yep. So, alpha helices, they can be, what is it? Hydrophobic, hydrophilic, and they can also be, beta sheets can also be the same. So know your definitions because she evidently asks questions kind of a little bit using big words. So we have to know the big words, the wordy words. Beta sheets can be anti-parallel, but alpha helices cannot. So
Shit, everyone.
So if you have anti-parallel, just kind of means that your alpha helices can go like this, and they can also go the same way. So they can sit next to each other in parallel like this, and they can also sit anti-parallel as well. And your beta sheets are much easier to see anti-parallel because your beta sheets will go like that because they're straight lines, whereas with alpha helices, it's harder to see it. So a lot of times people don't. But she does have that as a point, I think, in her lecture, that both of them can be parallel or anti-parallel. I think it's a bit specific, but it's nice to know. What two molecules bind together in an alpha helix and a beta sheet? Which orientation are they in? So this is your phi and your sigma bonds, if I remember, or your, I believe. So do you remember which one connects to what? So if you have a beta sheet and an alpha helix next to each other, first off, what kind of bond is that can't keeps them together? So that's kind of the breakdown of this one.
Can you go over this one?
Yeah. So this is about hydrogen bonds, if I remember correctly. Hydrogen bonds. So your hydrogen bonds to your oxygen. So this one's kind of a bit of a tricky question because there's a lot of words, and this is what I was talking about, that the person who wrote this, like, they definitely like really gave you guys a lot of headache.
Why is it
O to H. So it's a hydrogen bond. So first off, that knocks out these two because there's no hydrogen. And your hydrogen bond always does to oxygen. So you have oxygen to hydrogen in this one. Yeah, it's the carbonyl oxygen.
Wait, no, they're both wrong. Because it's the carbon on the carbon. It's the carbon, I'm sorry, it's the oxygen on the carbonyl, The oxygen on the carbonyl
So
and the nitrogen on the amine.
if I remember correctly, there's
I'm
in
reading it from the notes.
in the beta sheets, there's bonds like that in between it, if I remember correctly. So these are going onto one beta sheet. This is another one, and that's how you end up with the beta sheets sticking together, is these hydrogen bonds.
So you can recall, or I can read it here too. Similar hydrogen bonding. Similar hydrogen bonding in the alpha helix, but where the carbonyl oxygen of one strand bonds to the amide nitrogen of the
Hydrogen?
similar to the hydrogen bonding. So in the alpha helix, it would be hydrogen bonding, but in the
Yeah.
beta sheet, it's the carbonyl of to the amide.
Wait, I'm not trying to, like, who wrote the question? I'm not trying to call you out, like, I wanna know what their reason, like.
I believe Adams wrote this question. She's not here.
I'm calling anyone, I just wanted to know how they found it.
But
I just wanted to say that I think it's just mixed up. I think it's just a typo, but
I can double check on the word, but this is why I emailed Dr. Linger, is because there were a lot of questions that I got, and I wanted her to double check.
She did, I mean.
I may have accidentally like misprogrammed it too. That's
She did
also a
read it.
problem.
I would just email her or even go in there. I know like it's after our quiz, but at least for the test, I would go in and ask her, but she did look at that. And she did.
Yeah.
I don't know why she didn't email it.
Yeah.
That's
So
She went all over the place.
I'll say she probably was reading it fast and saw that the first part of that was right. Just read it from the notes that she literally put on the slide.
yeah. Yeah. So I didn't misprogram it in there, but I think that that's what she's getting at, is that that's hydrogen bonding. So I think that that's what she's getting at. All right. The interactions between beta strands are interstrand, true or false? We're more than halfway. So are they only intrastrand or not? They can be intrastrand or extrastrand. They can be both. That's what this question.
They didn't say phrase it right.
Yeah. So sorry. All right. The interactions between the alpha helix are intrastrand. So I think that this is from a particular picture in the slides. So to make that alpha helix, it has to bond to itself, whereas the beta sheet doesn't have to have that. All right, this one's a really particularly hard one, if I remember correctly. Whereas this one's like, what's the phi bond? And I think the other. Yeah, this is what the one I thought that we were getting earlier. Okay. Yeah, what slide is this from?
I'm thinking about, is this slide 13?
I think this was in this slide.
So is this right or wrong? Sorry. Okay.
The phi bond is the nitrogen oxidation.
Okay.
Psi bond.
Okay. Hey, there you go. What slide?
Slide 9.
Oh, okay.
Know your phi and your psi bonds, maybe? I think this is pretty specific. All right, anomers of the sugars are differentiated by the position of the hydroxyl group on the anomeric ketone, the anomeric carbon, the anomeric hydrogen, or all of the above. Okay, so you guys know why, or yes, no? Okay, I definitely got that one wrong, so don't look at me for this one. I did not start into these ones. A simple sugar molecule has the molecular formula of CH2O to the nth power, CH3O to the nth power, CH3O2, or CH2O3. So yeah, your sugars are all carbons with 2 hydrogens and an oxygen. Which of the following sugars is common? So your triple carbon, your 5 carbon, or your 6 carbon sugars, or all the above. Got your fructose, glucose, galactose, maltose. So all of them can be common sugars. Yeah, it is a gotcha. All right. A monosaccharide missing 1 or more hydroxyls? I think that's what that was supposed to say. All right, your deoxy sugars. That's why they're called deoxys, because they don't have the hydroxyl, if I remember correctly. Which one? All right, so these are all the lipids. So, so with the lipids, I believe this is, you have to think, all right, what is your lipid bilayer? What is fat? I'm quite fat. I have a lot of, no. Can lipids be used in molecular recognition? So multiple, I think there are multiple answers. No, okay. Yeah, so which one is not one of the lipid roles? So is not a primary messenger. All right, so I have the structure of cholesterol here. Looking at that, do you think it might be hydrophobic or hydrophilic? Just looking at that. Yeah, but you have to look at, there's only one OH on there, and when, yeah, so does cholesterol get clogged in your bloodstream? So yeah, so no, cholesterol gets clogged in your bloodstream because it falls out of it stays on the side because it doesn't want to be in the solution of your blood because it is hydrophobic. So it's very sticky, very hydrophobic. All right, which one is not a type or transport across the biomembrane of the cell? One is not a type or transport across, yeah, the biomembrane of the cell. So which one of these does not happen? Is that right? Okay. All right. Yeah. So this is not. So you can have a passive diffusion of a very hydrophobic molecule like cholesterol across the membrane. And then you can have mediated diffusive transport. So you need a protein to help you across the membrane. Or you can have active transport like your sodium potassium pump. But osmosis is specifically with water, and water is a polar molecule, and your membrane is nonpolar. So you're not going to have transport across. So that's where that one comes from. So this one, what is this? This is what is the sterol? So this is the picture of the sterol. So what does it look like? So is it a long hydrocarbon chain? Does it have a carboxylic acid head? Does it have a glycerol backbone? Does it have a polymer of isoprene units? Or is it 4 rings fused with hydroxyls? So just made it really easy by putting the picture up there. So it is 4 rings. But there are other, so you have your carboxylic acids that are going to come up. So these are definitions of other things that we are going to have to learn later with lipids. So this is one of mine. Sphingolipids stiffen the lipid bilayer. They do. So just press true. If I remember correctly, they do. But yes. So you want your lipid bilayer to be enough fluid for stuff to move around, but like there are things that you don't want it being so fluid that it just like pops apart. So sphingolipids play an important role in making sure that that doesn't happen. All right, so this is our math question. So this is from, I believe, number 2 in the question. You have 120 seconds. So if a weak acid is 96% neutralized at a pH of 4.7, what is the pKa of the acid? And we have some paper here if you guys need paper. So I have it worked out so I can show you guys how this one is done in particular. I believe, oh, I thought that we had one more question. Maybe I didn't save it. So it says that there's 34 questions. Did I not save that?
It's supposed to be more than one minute, you're on there. So we're missing 6 questions. So I will page through the questions in a bit and show you guys the last couple questions.
So this is how you do it. So why you do 96 over 4 is that if you have a percentage, you're going to have that molarity directly. So you don't necessarily need to figure out what the molarity is. You already have the ratio. So you can just, but I just like that it turned into a log of 24. So very simple. So I worked this out so that all right, are we good? Or is Casey still writing down stuff? Good. Okay. All right, there's only one pKa in a molecule. There is, however, 6 other questions that didn't show up that I accidentally didn't save in here. 2 of which I do actually want you guys to actually see. The other ones I'm like, eh. They're okay. They're good, but I put them in last minute, so they didn't save. You can have multiple pKas on every single group that can deprotonate. So okay. So let me go back. Let me see. I'll show you guys this. So there are 6 questions that didn't get in here. So this one, 38. So a drug with a pKa of 5 passes through the stomach, goes into the duodenum. Where will it be protonated? Will it be protonated in the stomach or the duodenum? Duodenum. Stomach. Good. All right. So we have a patient present in the ER with an opiate overdose. Their blood is 7.3. We pulled a blood panel. We find that their blood pH is 7.3. What caused it? So let me see. So this is the one that I wanted you guys to see. So you have hypoventilation, you have alkalosis, or you have acidosis and hyperventilation. So with an opiate, I know for a fact that she's going to ask about acidosis and respiratory because this is something that happens in the ER quite a bit, actually. There are certain drugs that cause this.
How could she ask us?
Yeah.
Oh, is it oh.
So, no. This is not from, no. This is more, because a lot of the pH stuff is we're going to get, but she is going to, I think for a fact, if there was anything I would ask you guys if I was a professor, it would be about this stuff. So when you have hypoventilation, you have acidosis. If you have hyperventilation, you have alkalosis. So all those are out of the same class of prion and folding misproteins. So you have like Scrapie and Creutzfeldt-Jakob disease. They're all from proteins misfolding, and then they happen to misfold in such a way that they turn into enzymes that facilitate the misfolding of the other proteins. So it just starts to kind of cascade, and that's what it seems like Alzheimer's is. So this last question is, so let's say we get that, we figured out what it is, why that patient, their pH is a 7.3. This one's more of a therapy question. This is way out of your scope, but I thought that you guys would. So your kidneys will balance your pH, but only if it's like 7.41 or 7.39. Once you start to get past 0.45 or 0.35, then you have to actually intervene. And then if you're going, so is their blood acidic or alkaline? So yeah, so their blood is more acidic, so you want to put bicarbonate for them because bicarbonate is an alkaline solution. And then lactate ringers and saline might make them even more acidic. It'll bring them up to neutral, but it has the possibility of making them more acidic, possibly. So that's why those are not good. But it's a little bit outside, but I thought that those would be interesting ones to do. So do uncharged chemicals distribute faster in the body than a charged chemical? So yeah, they do. And I think that this is the last one, yeah, that I missed. So these are all positively charged, if I remember correctly, instead of negatively charged. All right, so that's it for this. So congrats, go home. So evidently